Hey biology students, this is a video over cells and their environment. It's called transport, so we're going to go ahead and move along. The first thing I want to talk about is the cell membrane. Now we've already had notes on this, so you should probably know what the cell membrane is made of. But I want to add something to it today. So you know it is 2 layers or a bilayer of phospholipids, heads out, tails in, facing each other, but we need to add to it a protein. So there are proteins floating in the cell membrane, kind of like if the phospholipids are the lake and the protein is you in a life jacket and you're just kind of floating around in the lake. Kind of bobbing up and down and moving around a little bit, but still suspended there in that layer. This is an actual picture of cell membrane, and you can see the double layer of phospholipids here. This is one cell, the inside of one cell, and this is its neighbor, the inside of another cell. And so you can see the cell membranes here. The fluid mosaic model is the model that we use to kind of draw out what we think the cell membrane, probably its function and also its structures. Before I go any further, I should have probably told you that you need to have a piece of paper out and you need to be writing this down. So you might rewind a bit and write down the first slide, and then you can kind of go ahead and write down the fluid mosaic model. So the proteins are not fixed, they can move around the membrane, just like I already said. It is selectively permeable, which means it is kind of like a bouncer, so it selects the things that can go in and out of the cell. And that's pretty much it for cell membrane. It looks in 3 dimensions, kind of like this picture, and I want to use the model for the rest of the chapter of this beaker. So here we have going from one phospholipid to a double layer of phospholipids to a 3-dimensional cell, which we would show like if the cell is in here and the beaker or container is the solution out here. So when we draw cells in this chapter, we're going to draw a circle inside a beaker, and this would be the intracellular space and extracellular space. Same thing as out here. We have some terms to define. The first term is solution, and I think this is review from last year, so I'll go quickly through this. Is a mixture where one substance is dissolved in another. A solute is the actual substance being dissolved. I like to remember it like the solute is what you add to the water or to the solvent, and the solvent is the stuff that actually does the dissolving. In most cases, it is water. So there are a couple of graphics here to help you remember solute, solvent, and solution. When we go ahead and draw the scenarios that we're going to draw for which way water is going to move, when we have a cell in solution, we might also draw it this way. So go ahead and copy this into your notes. We need a box with a circle in it where you can write a percent solute, percent water, and then percent solute and percent water outside. So this is the cell and this is the solution. Hopefully that makes sense. We're also going to put numbers in here. So I made these up. So if it's 5% solute, 95% water, it's going to equal 100 every time. The outside solution also equals 100%. The solute and the solvent both make up the solution. So 3 plus what equals 100%. And that would obviously be 97. So you can fill in the 97 for that question mark. And this is the format that we're going to use when we do these problems. The concentration gradient is what happens whenever there are different concentrations or different numbers of molecules inside a cell and outside of a cell on either side of the membrane. And so the way you can show this would be to draw, please don't draw this out like really detailed, but maybe just draw two lines like they're the membrane, and then you can draw maybe 4 molecules on top and 2 on the bottom. And that would be a concentration gradient because there is a difference in the number of molecules or the concentration on one side of the cell and the other. The opposite of that would be dynamic equilibrium, where we had maybe 3 circles, molecules on top, and 3 on the bottom. Now, those circles could be, they could be gases, they could be ions, they could be other molecules like sugars or salt or whatever it is, but the distribution on either side would be equal, so like 3 and 3, and that would be dynamic equilibrium. Now they're still moving back and forth. The dynamic part of it means that these 3 don't stay on this side and these 3 don't stay on this side. They're always moving in and out. They're just moving at equal rates so that at any given point in time there will still be 3 and 3. I hope that makes sense. There are characteristics of passive transport. There are 2 types of ways that things move. The cell moves things in and out of it. One of them is passively, and the other is actively. And so, as you can imagine, passive is easy, doesn't take any energy from the cell, and it just goes, it's just easy. It's what it wants to do. And then active we'll get to later, and it takes energy from the cell because it's kind of like going up a hill. But let's talk about passive transport. It is the movement of a substance that diffuses from high to low concentration until equilibrium is reached. And so let's draw over here. Go ahead and draw your membrane again. Just 2 lines is fine. So we might draw 4 or 5 over here and just 2 over here. And then our arrow is going to go from here to here. Okay? And so we're going to go in this direction. So we're going to go from high concentration to low concentration. It doesn't require any energy because it happens naturally. And just like rolling down a hill. There has to be a difference in concentration for this to take place. So maybe jot down all of those for passive transport. There are 4 types of passive transport I want to talk to you about. The first one is diffusion. I don't know why these particles are all equally distributed right now. They're supposed to start out like all in one corner and then they all spread out, but that would be diffusion. So the first type is simple diffusion. It's the movement of small neutral molecules from high to low concentration across the phospholipid bilayer. Examples of molecules that simply diffuse, which means they just go from high to low through the phospholipids, would be carbon dioxide, moving from high concentration outside the cell to the low concentration into plant cells, because plant cells need carbon dioxide. Oxygen, moving from high to low inside of our cells. And so, because we need oxygen. And they just passively move through. They're so small and neutral, and so they don't need any energy or anything like that. They just slip through the phospholipids. I think this is supposed to be an animation, but that's not happening, so maybe I'll show you in class. The second one is osmosis. This is just the diffusion of water from high to low. So diffusion, simple diffusion itself, could be the diffusion of any types of small neutral molecules from high to low concentration across the membrane. Osmosis is just the diffusion of water. So if there's more water over here and less water over here, water is going to move to where there's less, from high concentration to low concentration. There are 3 different situations for solutions. There are terms hypertonic, which means a solution with higher solutes. There's hypotonic, a solution with lower solutes, and isotonic, which is equal solutes. So go ahead and jot those down, and you will have those to use in class. The situations where different cells are in and will act differently would be these. So in a hypertonic solution, so the cell is hypotonic and the solution is hypertonic. Water will flow outside of the cell and the cell will shrink up. In a hypotonic solution, which has a lower concentration of solutes outside of the cell, water's going to move in. So the cell swells. And then if you put a cell in an isotonic solution, water will move in and out. It will stay the same size. You might want to draw these cells just like a rectangle and, kind of shriveled up, water going out, water going into this one with, the cell kind of big, almost ready to burst, and then isotonic is arrows in and out. Plant cells have cell walls to keep them from bursting, and we call the amount of water in the plant cell turgor pressure. So if there's a lot of water in the plant cell, it's exerting force outside of the cell wall, and it's nice and full and it'll stand up. But if it has low turgor pressure, just like these plants, it'll wilt. Okay? Here is the same as the last slide here. Here is the same type of scenario. Okay? So which hose has more turgor pressure? This hose or this hose? And the answer is, of course, this one. Which cell has more turgor pressure? This cell right here. So that's all turgor pressure is. It applies to plant cells. Animal cells, they do not have cell walls. So they need a different adaptation to help them pump water out if water is always flowing in. So they have contractile vacuoles. Some of them do. You can see them in this video. I don't know why this video works and the rest didn't. But you can see that the contractile vacuole fills up and then gets spit out. So here is the contractile vacuole that is empty, and here it is full. So it'll fill up with water, then spit it out of the cell, and that way the water won't flow in, and cause the cell to burst. Diffusion through ion channels is the third type. Let's review. The first type was simple diffusion, the first type of passive transport. The next type is osmosis, which is a diffusion of water. The third type is diffusion through ion channels, and that's just when we have ions that want to go from high to low. See, they're high here, they're going to low concentration. They can't fit through the phospholipids because they're charged. So we say any molecule that's charged needs a protein. It's like a special door to go through, and these are called ion channels. These ion channel proteins. So sodium, potassium, chloride, calcium ions all need to go in and out of the cell, and they need proteins to do it because they cannot slip through the phospholipids. So diffusion through ion channels. And the last one is facilitated diffusion, which is not a charged molecule, but a large molecule. So I kind of like to say if it's large or charged, it has to have a protein to move it through. We're still moving from high to low. It's still passive, but we just can't get through this door to the phospholipids, so we have to have a special door. So we call these carrier proteins because they carry large molecules inside and outside of the cell. So if you write this down, uses a carrier protein to transport molecules. Go ahead and record this flow chart here to kind of review what we've done. We talked about transport. And then there are two types, passive and active. The types of passive transport I just went over in order would be simple diffusion, and then osmosis, ion channel diffusion, and facilitated diffusion. So that's all the notes for passive transport. Next would be on to active transport, which we will cover another time. Good job, guys.